Well, hello everybody. I'm Matthew Walker from Sartorius, based in the UK, and welcome to the latest in our series of webinars on the ambr bioreactor systems. Today our focus is going to be on the ambr15 system, and you're going to hear from two fantastic speakers today. Javier Lobo is an application specialist. He works for Sartorius, based in Spain, and he's going to give you an in-depth overview of the ambr platform and some other related Sartorius products. Our main speaker today is Marta Ruiz from 3P Biopharma, again based in Spain, and she's got a very interesting presentation on how they've been using the ambr15 in their research. Got 4 or 5 housekeeping points first, though. We expect the webinar to be about 40-45 minutes in length, and then we have a chance for a small Q&A at the end. So on your screen, you'll see an Ask a Question feature. So please, at any time during this webinar, if you have any questions, please type them in there, and we will try and deal with as many as we can at the end. If it's very technical or requires a much longer answer, we will get back to you kind of one-to-one offline. But we will share all questions, all answers with everyone who's registered for this webinar. This webinar is being recorded, and we will have a recorded version available next week, and you will be able to listen again or indeed share this webinar with colleagues. They would just need to register as well. If you experience any technical problems, there's an event support button on your screen as well, and if you click on that, our webinar partner, Workcast, someone in their tech support will get in touch with you and we'll be able to assist you. So that's about it from me. So I'm going to hand you over to our first speaker now, which is Marta. So Marta, over to you for the first introduction. Thanks.
Okay. Thank you, Matt. Hi, everyone. So we are going to start the presentation. The agenda that we are going to be following during the presentation is going to start with a brief introduction about 3P Biopharmaceuticals. It will be followed by a short introduction regarding the ambr15, and then we will go through the case study regarding the high-throughput screening. First, the results related with the screening of the media, following by the screening of the feeds, related also with some supplements screening and the addition strategy study, following by a verification steps, and then we will have a part for questions. So 3P Biopharmaceuticals, we are a leading contract development and manufacturing organization. We are based in north of Spain. We offer personalized services covering all needs related to process development and GMP manufacturing of biologics and cell therapy. Although our core expertise are biologics, we have an exclusive area with separated rooms from cell therapy unit. We have experience working with both new biologic entities and biosimilars. This includes various products such as recombinant proteins, monoclonal antibodies, and fusion proteins. Quality is in the heart of 3P, and we are truly committed to offering the highest quality solutions. Thus, we work closely with the agencies, and we are GMP certified for biologics manufacturing, and we are also have the GMP certification for some cell therapy products. Moreover, we are working on FDA certification, and we are supposed to be audited by 2018. This will be an important milestone for 3P because this will permit both our clients and our company to the high market to the United States. We have a unique goal, and it is making the product a success. We have built a leading facility designed able to offer solutions in every step of product life cycle. We are providing solutions starting from the development all the way to product commercialization. For the cell line development, our top-level team is researching the best expression system to support your biologics program in the most efficient way. We perform not only RCB, but also GMP cell bank manufacturing of master cell bank and working cell bank. During process and analytics development, we always think ahead with the goal to achieve best yield, cost of goods, time, scalability, robustness, and attractive licensing conditions. What is also important, we are extremely experienced in technology transfer and any stage of product lifetime. A dedicated technology transfer team handles process and analytic transfer, assuring the seamless transfer, implementation, and target protein manufacturing. Additionally, a scale-up of development and transfer process is executed in an effective and experience-driven way. We are not only providing materials for clinical trials, but also manufacturing commercial batches, drug substances, and drug product release. At advanced stages of product development program, we are supporting our clients with process characterization and validation, which has become our core expertise. Our process development area is driven by an industrial vision and supported by team, equipment, process being aligned with cGMP manufacturing methodologies. We are always thinking about the future of the program, thus we are developing cost-efficient, scalable, and robust processes. This way of working assures a seamless transition of each process from the development to the cGMP manufacturing plant. Thanks to a unique combination of the latest technology and equipment with know-how and team spirits, we support product development in a high-quality and cost-effective way. 3P Process Development Team occupies modern and well-equipped laboratories. We understand the time, cost, and quality pressures. We are committed to meet the needs and providing the best tailored, flexible, and cost-effective manufacturing solutions. As a robust, both mammalian and microbial manufacturing partner, we combine our knowledge, experience, and state-of-the-art equipment for your biologics manufacturing. 3-piece scale-up and cGMP production fermentation cell culture volumes provide an optimized and cost-effective manufacturing solution for each product. So with this, I now give the word back to Matt.
Thanks, Marta. That's great. Thanks for the introduction. Okay, I'm going to hand over now back to Javier, who's going to give you an overview of the ambr15 product and some related products. So over to you. Thanks a lot.
Thanks, Matt. Okay, thank you very much. We are starting now with the overview of the ambr15, which is the system has been used for the case study that Marta later on will present. Okay, in Sartorius, we have not only the ambr15 technology in single use, we have plenty of possibilities for scaling up from drug discovery to production. So the ambr15 is a system, the system with the capacity of 50 milliliters working volume, but also we can move to the ambr250, which is a system with the possibility to run experiments with 250 milliliters, especially for process development, but afterwards we can scale up in single-use technologies from 50 to 2,000 liters, as you can see in the presentation. The good point in here is that we can have a very nice scalability approach because we are keeping the different, let me say, ratio of construction of the system in terms of agitator, but also in terms of physical components. So the next slide, you will see in deep the ambr15 bioreactor family itself, and then we have two possibilities. We have the ambr15 cell culture, which is dedicated for eukaryote cells, but mammalian cells. The system can manage bioreactors from 10 to 15 milliliters. I mean, 10 is the minimum working volume, 15 is the maximum. And then we have the station with two possibilities. One of them with 24 bioreactors in parallel, but also we can have the possibility to expand to 48 vessels. We have another version of the ambr15 fermentation, more dedicated for bacteria and MO application, and then the working volume changed a little bit because the specification of the cultures needs more head space for the application. Then we have maximum volume of 12 milliliters, but the minimum is 8. In this case, the station can work in parallel with 24 vessels. Both are working totally automated, and obviously the system can be managed by software, dedicated and tailored for the application, and of course the system can manage in parallel processing in totally disposable micro bioreactors, because the bioreactors comes already gamma radiated, already sterilized, and then ready to use. When you finish your experiment, then you throw away the vessel and then you can start a new experiment immediately. How is the cell culture workstation ambr15? You can see in the presentation we have a robot. The robot is manage all the fluid automatically and can be programmed by the computer. And then the system is for caps, lids, and liquids. I mean, to manage all that kind of things. So we have obviously in the console the 2 times 12 bioreactors, total 24 in this version. It means every 12 bioreactors can be run with independent temperature and steering control. I mean, pack of 12. In the other part of the system, you can see the place for the tips. We can have 1 milliliter, 5 milliliters, different possibilities. It's just an example, but also we have the media, the inoculum cells, and any kind of region necessary to control the system. All the system can work with pH and DO measurement and control, and every single vessel is totally independent to measure and to control both parameters. Optionally, we have the possibility to have a tap-line pH measurement, I mean, just to make an external system in order to make the calibration or recalibration during the process, but also we have the possibility to integrate cell counting for viability system, I mean, measurement system, but also integrated measurement of multiple analytes with the possibility to be integrated in the system in order to make different strategies. Now, regarding the vessel, I mean the microbioreactor, the 15 one, we have the 50 milliliters vessel in here in the left part, but also in the right part you have a classical reusable glass vessel. You can see that both, I mean, in the case of the 15 is a mimic copy of the existing, let me say, classical vessels. So we have feeding port, sample port, we have impeller drive, we have a spark gear in order to make the gassing, but we have the pH and DO2 sensor integrated in the system, but different kind of impellers, 3-blade for mammalian, but also we have the possibility of different Oraston. We are working with MO application. So we can say the ambr15 vessel is really a real bioreactor with the full control that you can find in bigger vessels. Application for the ambr15 cell culture, the most, let me say, typical and valuable application you can see in here, like clone screening. Obviously, we instead to have the typical shake flags, we can use our ambr, which is much more cost, let me say, effective, but also is really convenient in terms of control, because we have really independent control of every single vessel, and we have all the parameters and variables with measurement, but also with control. Obviously, for process development, media development, also we can make perfusion, because the system can be programmed in order to keep the cells inside, but also to make media exchanges, transient transfection. Also, we can have with microcarriers, instant cell expression, but also with stem cells and different cell lines. So the system is really ready to run in different applications without any problem, comparing with, let me say, the typical technologies on the market. On top of that, of the ambr, we can offer another service, which is the CHO-Optimizer. The CHO-Optimizer, according to design of experiment, means that is a service that we can offer with the ambr. In this case, one specialist from media, for cell culture media division, can support the experiments in 3 different ways. So the customer can select what is the best for them. For example, in the case of module 1, you can see in here, we can supply different media mixture, normally standard, based in the standard media, according to design of experiment, in order to looking for the best, let me say, media, or the best mixing media, and then we can measure the metabolites in order to understand exactly what is the best media for our application. In the module 2, we have design of experiment of the selected predicted media from the module 1, and then we add the process parameter in order to optimize the process itself. And on top of this, we can make also media analysis over the media, the metabolites already done before. And in the module 3, we can make design of experiments approach also for the feedings and feedbacks. I mean, we can add different supplements, different agents, different reagents in order to looking for the best, let me say, productivity for us. So it's a service offered for us when the customer needs, for example, to be faster and to looking for the best solution in a shorter and cost-effective way. Then summarizing why I must to choose the ambr15, it's clear we can increase the capacity and productivity with totally automation approach and single use. So the benefit is clear because less materials is necessary to use. We reduce the labor and then we can increase the output. I mean, the number of experiments in a very short time with very, very robust approach. So it's also a predictive performance because it enables good decision in the beginning because you have the safety to use all the time one new vessel for every single experiment, so you have not problems anymore in terms of cross-contamination or problems with your, let me say, probes and so on. So the reality of the results are really good in order to make later on and the real scale up in the right way and the faster way. The ambr15 is offering to you high throughput possibilities combining with the design of experiments. In this case, you can start from the real beginning of your process quality by design approach in order to use later on for your, let me say, scaling up, but also because the regulation and the understanding of your process will be faster, and then to have the product to make to clinic phases and but also commercial will be much better and faster. Obviously, the system is something which is a standard today. It's an industry standard chosen by more than 90% of the top 20 biopharma companies worldwide. Then there are plenty of different articles, different posters, bibliography available to you, and we can share different, let me say, experience and experiments done for that companies when it's possible. Finally, we can say the ambr15 is totally part of an integrated upstream platform from cell line to manufacturing because you can understand exactly your process from the beginning and then using all that data for a faster speed to the clinic. Okay, from my side, that's it. Matt?
Thank you. Thanks, Javier. Great information. See a few questions already being posted. So if you have any product-related questions, technical questions for Javier, please don't hesitate. Get them down, and we will try and deal with as many as we can at the end. Right. I'm going to hand over to our second speaker again. So Marta, over to you now for the final session.
Okay, thank you. So we are going to focus now in the case of a study. See, in this case, we have the challenge to produce a monoclonal antibody. The cell line that we were using in this case was CHO-S cell line, and our goal was to produce high protein title in a good protein quality. In order to get the best culture conditions for this cell line, we planned the high-throughput screening, both media and feeds, so we have all the options tested. First steps were focused on media screening, and once we have the media defined, we follow with the screening of different things. So the study started, as I said, with the clone already selected, and the first part was regarding the screening of the media. In this image, it is represented the media screening procedure that was followed, and we did in total 3 runs at this stage. In the first run, we did a screening of different commercial media from different suppliers. From this first screening, 2 best media were selected, which were used for the second and third round of screening. From these 2 best media, the suppliers provided us a wide variety of customized media to be tested. So the second round of media screening was composed by 9 different media supplied by the supplier number 1 that were tested in the second round. In the third round, it was done with 16 different media provided by the second supplier selected. I have to say that in each round of screening was done in batch during no more than 12 days. The ambr innovative technology allows us to perform the screening with controls, duplicates, and different conditions at the same time. With the current technology, the alternative to ambrs would be the shake flask with the limitation that implies, such as absence of process controls, higher variability between flasks, so that more controls, more replicates, and therefore more times will have this required. The results obtained from the first round of screening are presented in the following slide. So regarding the viability cell density that is represented in the top graph, the top left graph was similar in all conditions with a slightly fluctuation during the batch, reaching a maximum of around 5 million cells per milliliter. Regarding the viability, all cases saw a similar tendency, decreasing during the batch as expected. Taking into account the protein title that is represented in the two lower graphs, the productivity was not very high and the two best conditions are indicating they were media 1 and 2, which were chosen for further analysis. From this first round of screening, the two best media were selected, as I said, and explained previously. So the second round was done with one of these suppliers, in this case media 1, with different customized media provided by the supplier. The viable cell density in this case was slightly higher in this round, reaching a maximum around 7 million cells per milliliter. The decrease in viability was also shown in this batch, as you can see in the upper graph. And regarding the productivity, the amount of protein obtained was similar than the results obtained in the previous screening. So we didn't observe an improvement in any of the new media tested in this case. So far, the best media that we have seen is media 5 in this case, that correspond to media 1 that we saw in the first media screening. The third media screening was done with different customized media coming from the media 2 selected in the first screening. In this case, the data obtained regarding the viability cell density was similar to the second round of screening, around 7 million cells per milliliter, although the productivity was much lower compared with the media tested previously, as you can see in the graphs represented in the low part. After the 3 rounds of screening, in total I have to say that we test 32 different media in a period of a month. The 2 best media were selected for further analysis, and the selection of media and feeds has been done depending on cell density, the protein title, and the protein quality, defined by glycan analysis, the activity of the protein, change variants, and also aggregates. Further steps that we follow in the optimization of the production of this monoclonal antibody was the screen of the feed, supplements, and feed strategy. It is represented in the next slide. So here we have it, and we are going to follow different steps during the presentation that are globally called field screening, but it is composed of different parts. The first one is related with the feed screening, in which different ambr batches were done, testing 15 different feeds in combination with 2 best media previously selected. In this part, in totally, we test 48 different combinations. Once we have the media and the feed defined, we move forward for the next screening. In this case, it was focused on supplements. We added to the media during the batch, and in this case, 37 different conditions were tested. The last screening was done focused on the strategy of feeding addition. In this case, 24 different addition strategies were studied based on feed concentration, days of addition, and amount of feed, and different characteristics were tested. Once the process was defined, 1 media, 1 feed, 1 supplement, and the feed strategy was defined. So we did a verification step in 1 liter benchtop bioreactor, following the parameters that we defined in the ambr with the previous screenings. So following the scheme, we are going to present the data from each part. We are going to begin with the feed screening. In this part, I have to say that due to the high amount of data that we have, we are going to present only the best, the worst, and the media data in order to simplify the presentation. The feed screening was done in batches that were during around 14-16 days. The amount of viable cell density observed in this feed screening was higher compared with the viability cell density that we were achieving in the screening of media. We were now reaching around 13 million cells per milliliter in the first feed screening and around 14 million cells per milliliter in the second round of screening, as you can see in the left graphs. Regarding the protein titer represented in the right graphs, in the first screening we were obtaining a productivity around 2.5 milligrams per milliliter in the base case, and in the second feed screening we were obtaining around 3 milligrams per milliliter, also in the best combinations. Taking into account the viability cell density, the productivity, and the quality of the protein, we did the selection, and now we move forward with 1 media, 1 feed already selected. So we keep on doing the further analysis. In this case, the next part was focused on supplements added to the culture media in order to improve either the quality of the protein or the productivity. 37 different conditions were tested in this case, so a lot of data were collected, and as same as before, in order to simplify the presentation, again, we are going to show only the best, the worst, and the media data as a representation. In this case, we have represented some conditions of the supplement screening together in the same graph, as you can see in the presentation. Regarding the viability cell density, the amount of cells in this case that we were reaching, it was up to 14 million cells per milliliter, and the productivity was also better, reaching more than 4 milligrams per milliliter. After each screening, we were observing an increase in the productivity, and the protein quality of the selected conditions was also appropriate and compared and similar to the standard. So far, we have one media, one feed, and the media supplemented defined. So the further analysis were focused on the strategy of the feed addition that we could follow for the scaling up. As shown in the scheme, different conditions for the feeding were tested. In this case, 24 different conditions were studied related with the addition strategies, feed concentration, days of addition, and amount of feed. The viable cell density observed in this case were similar with the best options obtained previously, and regarding the protein title, we saw some difference between the best and the worst conditions, but the best productivity was around 4 milligrams per milliliter. These conditions allow us to know the best options for feeding strategy, focusing in the scale-up. At this point, we have the media, the feed, the media supplement set, and also the feed strategy optimized to a better and good quality productivity. We proceed with the verification step and in a scale of 1 liter. So we are arriving to the last part of the presentation. We consider the verification step as a performance of the conditions set with the ambr, but now in a 1 liter scale benchtop bioreactor. In this case, we were using 6 bioreactors in parallel with the, as I said, with the conditions determined previously with set in the ambr. In the graph are represented the 6 bioreactors, and also we have included the data coming from the ambr as a control. And as you can see, we saw a very similar profile, either in viability cell density and also regarding the protein title. This shows that the robustness of the screening and development done in microreactors has been very consistent. So in the results that we found here. So as a summary, we have included the last graph, which shows the evolution in protein titer during the screening and optimization that has been performed. The increase in productivity is clear. We have seen an increase in 4 times the initial title obtained from the first screening that was done. And once the media and the feed was selected, as you can see in the graph, we didn't see a significant improvement regarding the productivity. So as a conclusion, we have to say that we tested 128 different 128 different batches in several replicates within approximately 5 months. The ambr system facilitates the possibility to test many different conditions in parallel, saving time and money. And the protein title, as I said, was increased 4 times along the experiments. And the conditions and results obtained with the ambr system, we saw that they were reproducible in a 1 liter benchtop bioreactor. So here is our contact in case you want to come up with us for anything. And yeah, the last part is coming if there is any question.
Okay, thanks, Marta. Thanks again for a wonderful presentation. So anyone, if you're listening and you have questions for Marta, you've got a couple of minutes if you want to post a question, and we'll try and deal with it. We've had quite a few questions coming through now, so I'm going to throw a couple to Avia first. So first one is on analytical systems. So could you tell us which analytical systems is it possible to actually connect to the ambr15, particularly for at-line sampling, Javier?
Yeah, sure, Matt. Yes, there are a number of analytical instruments that can be integrated with the ambr15 for automated cell counting, cell viability, pH, and metabolite analysis. These include, for example, the BiCell, CDX, also our ambr analysis module for pH calibration, but also the Nova Bioprofile Flex 2 analyzer. This is the more complete. So for any other types of analysis samples, the samples can be replaced in tubes, plates, by the liquid handling robot, and then you can take for offline analysis by the operator. But in general, yes, we can use a different analyzer. We can integrate that analyzer into the system. Okay.
Okay. No, thank you. Second question for you, Javier, is one on pH, and it's a question we often get. But could you just briefly describe how pH is controlled with the ambr15?
Yeah, sure. In the ambr15, every single microbiorreactor has a dedicated line. So in this case, for the CO2, we are introducing the CO2 in every single vessel to control the pH. So there are independent gas line for every single vessel. So the control is totally done for without any problem, because we have the single-use pH measure integrated in the vessel, and then we can control by the control loop integrated in the software what amount of CO2 is necessary to control the pH. Obviously, if it's necessary to add base, then the robot can do it, taking, for example, from the region part of the console, any volume necessary to control the pH too. So it's really totally possible to control independently the pH in every single vessel.
Okay, thank you. That's great. Okay, I'm going to go to Marta now. There's two questions for you, and again, they're quite frequent questions that we get actually often. First question is, could you just briefly describe the method you're using to quantify the MAB titer?
Yeah. We mainly we use the protein A measured by HPLC. But recently in our facility we have implemented a new equipment. It is called Octet. It was with a protein A sensors that can be loaded in a 96 well plate. So it allow you to measure many different samples faster and in faster and less cost. So yeah.
Okay, no, that's great. And then there's a kind of a follow-up question. So in addition to the product titer, did you actually study some other product quality attributes? And if so, which ones did you think were the most important?
Okay, thank you. Yeah, in this case, regarding the product quality, we were also analyzing the glycan profile, protein activity as well, change variants, and also the aggregates.
Okay. In fact, I'll move to a third question for you, Marta, if you don't mind. And again, that's sort of another interesting question, but we do often get is, in your experience, running the experiments you were running, how many people did it require to sort of run this project and set up the ambr system?
For the ambr manipulation, in this case, we were one or two full dedicated people for controlling the equipment during the performance of the batch. And the time of the operator, it can depend on the analytics of the activities that are programming for the day, but it can be around 7 to 6 hours. In our case, we don't have the analytics implemented with the ambr, so we have to do the pH control, the cell counting, and also the metabolites analysis. We do it offline, so that's why for us it takes a little bit longer, because we don't have the equipment implemented in the ambr. So. But yeah, we do it like this.
Okay, and I'll do a fifth question back to Javier. Javier, a lot of people ask, what evidence have we got of this scale up? Is there anything you can talk about or point us to that would show how the ambr15 is scaling up to maybe 1 liter or 3 liter, 5 liter, whatever? Could you talk around that for a second?
Yes, Matt. Yeah, the ambr15 cell culture has shown comparable cell culture performance and productivity, but not only in the 1 liter bioreactor, as Marta mentioned during the case study. Also in larger scales, from 2 liters even until 15,000 liters in manufacturing scale. I mean, we have plenty of reference to share, but we are speaking obviously about the performance of the cell culture, but also the productivity. We are not speaking about the full scaling up regarding physical parameters, but in case of the full culture performance, but also the productivity, yes, definitely the system is totally comparable. The result coming from the ambr15.
Okay, we've got sort of 4 or 5 other questions that I'm not sure we're going to have time to answer fully here. There's some questions on perfusion and a few other things, which we will deal with that offline, but we will get the answers to any of these questions back to all of you. Remember, these slides are going to be made available to you next week, so you will be able to get the contact information and all the other kind of information from these slides that you may want, and you can, of course, share them with anyone else you wish. I'll just ask my 2 presenters if you could just have a quick look at the last 4 or 5 questions that are there in text form. If there's anything you feel you would like to answer, please go ahead now. Otherwise, tell me no, and we will move on to the next phase.
That's it from our side today.
Okay, so I think that's going to be the end of our webinar today. Thanks very much for everyone for contributing with your questions. We will get answers back to everyone who has requested an answer. Thanks once again to our 2 speakers in Spain. Great job, very comprehensive presentations, and we look forward to our next webinar, which is going to be probably in June this year on the ambr system. Thanks very much indeed, and have a good day, everybody. Thank you.
Thank you.